Hello students, welcome to EPG Pathshala. I am Dr. Nidhi Gar from Deshbandhu College, University of Delhi. Today we are going to talk on module RNA interference from the paper Molecular Genetics. The following are the learning outcomes of this module. You will be able to know how the phenomenon of RNA interference works. You will be able to learn about the various mechanisms of RNA interference. You will be able to evaluate the importance of RNA interference in regulation of gene expression and know the current and potential applications of RNA interference. And in the end, I would summarize the module. RNA interference, also known as RNAi, is defined as a mechanism or a biological process where the RNA molecules interfere with gene expression, thereby inhibiting it by destroying the specific mRNA molecules. The RNA interference is mediated by double-stranded RNA, which is clipped into smaller RNAs by the enzyme dicer. These small RNAs then associate with several silencing effector complexes, which then attach to homologous sequences of their target RNAs or DNAs by base pairing. These RNA molecules, for the purpose of inhibiting gene expression, share sequence homology with the gene in question. On the basis of protein composition of the effector complex as well as the nature of target sequences, these RNA molecules can lead to either mRNA degradation, translational repression, or genome modification. All these three processes ultimately lead to silencing of the gene expression. The act of repressing both transcription and translation is known as RNA-mediated gene silencing. As you can see in the diagram towards the right, the long double-stranded RNA molecules come in contact with the nuclease known as dicer. This dicer is present within the cytoplasm of cells, which cleaves these longer RNA molecules into smaller molecules, which could either be siRNAs or the microRNAs, which we will subsequently talk about, which then associate with the effector complexes known as RISC. And then, depending upon the sequence similarity to their targets, they mediate either the degradation of mRNA or the translational repression or genome modification, ultimately interfering with the gene expression. Now, if we go a little bit into the historical background of RNA interference, several, or I must say most of the biologists all over the globe came to know about RNA-mediated gene silencing in 1998, only after its discovery in Caenorhabditis elegans by the work of Craig and Fire, where a double-stranded RNA initiates sequence-specific mRNA degradation. But the roots of RNA interference dates back to late 1980s, at the time when the botanists across the globe were struggling with strange cases of gene silencing in transgenic plants. At that time, the RNA molecules were commonly used for bringing about a reduction in the gene expression in plants for several years. The single-stranded antisense RNA molecules were simply introduced into plant cells, which subsequently hybridized to the corresponding single-stranded sense mRNA. In the early 90s, the researchers were trying to alter the flower color of Petunia albas by introducing multiple copies of the gene encoding the enzyme chalcol synthase. Usually, the color of petunias is pink or violet. The researchers wanted to increase the intensity of the color such that they could obtain darker color flowers, and the enzyme chalcol synthase plays an important role in flower pigmentation. So they thought by injecting multiple copies of this gene, they will be able to enhance the color. But on the contrary, instead of getting darker colored flowers, the overexpression of charcoal synthase gene was not observed. Rather, they got less pigmented or fully or partially white flowers. This simply meant that there was a reduction in an activity of charcoal synthase. In fact, these white flowers were a result of downregulation of both the endogenous genes and the transgenes. Now, if you look at the diagram above, the flower on the extreme left is extremely darkly pigmented, which is the flower which is the original color. This is the petunia flower with its original pigment. The one in the middle as well as towards the right are those transgenic petunia flowers in which multiple copies of the gene charcone synthase was given and there was inhibition of gene expression. The researchers found that the downregulation of charcoal synthase gene was caused by post-transcriptional inhibition of gene expression or PTGS due to high levels of mRNA degradation. This phenomenon was known as co-suppression of gene expression, but the underlying molecular mechanism responsible for this was not known. A related phenomenon known as quelling was observed in the filamentous fungus Neurospora crassa, but the details were not worked out. In 1994, RNA-directed DNA methylation was discovered in transgenic tobacco plants, which happened to be the first reported case of RNA-induced modification of DNA. The next development was that the plant RNA viruses could be both initiators and targets of post-transcriptional gene silencing. In 1997, PTGS was reported to protect plants naturally from virus infection. Thus, by the year 1998, when RNA interference was reported, the plant scientists had already documented the following. First, a sequence-specific RNA degradation, also known as PTGS. Secondly, a sequence-specific DNA methylation that triggered TGS. Thirdly, RNA-directed DNA methylation. And fourthly, they proposed models for PTGS involving double-stranded RNA. After working with plants, the researchers set out to look for similar phenomenon in other organisms as well. The phenomenon of RNA interference was discovered by Craig C. Mello and Andrew Fire, for which they were awarded in 2006 the Nobel Prize in Physiology or Medicine. They were the ones who coined the term RNAi, and their pioneering work was published in the journal Nature in 1998. In 1998, double-stranded RNA was shown to cause gene silencing in other plants and animals. In 1999, scientists studying PTGS in transgenic plants detected the small 25 nucleotide long RNAs involved in mediating gene silencing. In the year 2000 to 2001, the researchers working with Drosophila demonstrated that short interfering RNAs, known as siRNAs, were 21 to 23 nucleotide long and were actually derived by the cutting or the slicing of longer double-stranded RNA that were capable of guiding mRNA cleavage. In 2000 itself, the nuclease known as RNA-induced silencing complex, known as RISC, was discovered, which associates itself with small RNAs and performs cleavage of target mRNA. In 2001, the enzyme known as dicer was identified, which cuts double-stranded RNA into short RNAs. In 2001, several natural small RNAs ranging from 21 to 25 nucleotides were isolated first from worms and flies and then from plants and animals. In 2003, small-sized RNAs known as microRNAs were identified, which arise from dicer processing of double-stranded RNA, which are then incorporated into RISC. miRNAs or the microRNAs are the key regulators of both plant and animal development. In 2003 itself, the relatedness of different phenomena, namely RNA interference, PTGS, and quelling, was established through genetic analysis in worms, plants, and Neurospora, which led to the identification of common components involved in the respective silencing pathways. Till then, these were considered to be separate phenomena. PTGS is now believed to be an equivalent of RNAi in the plant system. In 2002, the discovery of RNA interference-mediated heterochromatin assembly in fission yeast established the principle of RNA-guided genome modifications. This pathway involves the use of short RNAs produced by dicer and other RNA interference components for methylating histones. This generates condensed, transcriptionally inactive regions called as heterochromatin, such as centromeres. In 2003, RNA interference-dependent heterochromatin pathway was discovered in plants, whereas in 2004, a similar discovery was made in Drosophila. We will now talk about Craig Mello and Andrew Fire's classic experiment, which led to the discovery of RNA interference. They chose the model organism, the roundworm Caenorhabditis elegans, for their work. At that time, most of the work centered around the use of single-stranded RNA to bring down the gene expression, but they also wanted to test the possibility of interference by a double-stranded RNA molecule. So, they took two experimental setups. In the first experimental setup, they injected the roundworm with single-stranded RNA fragments, and in the second setup, they injected the roundworm with double-stranded RNA fragments. These single and double-stranded RNA molecules were homologous to the gene UNC22. This gene codes for an abundant but non-essential myofilament protein. Now, the expected outcome of this experiment was the downregulation of gene expression upon injection of single-stranded RNA. But an unexpected surprise outcome was a 10 to 100-fold decrease in the expression of UNC22 gene upon injection of double-stranded RNA molecules, thus proving that double-stranded RNA is even more effective in bringing down the expression of the UNC22 gene by mediating degradation of the resulting mRNAs. Injection of UNC22 single-stranded RNA into adult animals produced a modest effect, while UNC22 double-stranded RNA produced potent and specific interference and phenotype. Both the injected animals and their progeny showed behavior similar to loss-of-function mutation in UNC22 gene. The presence of double-stranded RNA initiates the degradation of the mRNA provided the mRNA has a sequence complementary to one of the strands of the double-stranded RNA. Few molecules of double-stranded RNA are required to initiate degradation of large amounts of mRNA. Now coming on to the molecular mechanism of RNA-induced gene silencing, the nature has evolved an efficient mechanism for gene silencing through the use of small 21 to 25 nucleotide long non-coding RNAs that are double-stranded and bind to the target RNAs in a sequence-specific manner. There are three main classes of small RNA, namely the short interfering RNAs known as the siRNAs, secondly the microRNAs known as the miRNAs, and thirdly the pi-wi interacting RNAs known as the piRNAs. Although these three RNAs arise from different sources, their mechanism of action is quite similar. The microRNAs are short RNA molecules transcribed from the cell's own DNA within the nucleus but are never translated into proteins. The DNA sequence that codes for microRNA gene is longer than the microRNA itself, as it includes the microRNA sequence and its reverse complement. The short interfering RNAs, also known as the siRNAs, are derived from longer RNA molecules which are linear, double-stranded and located within the cytoplasm. These siRNA precursors arise from within the cells from the following sources. They may arise from RNA virus replication, transcription of cellular genes, self-annealing transcripts, expression of transposons, experimental transfection. siRNAs mediate RNA interference by endonucleatic cleavage of their target RNAs. RNAi may be a mechanism through which cells recognize the viral RNAs and inactivate them, thereby providing the organism with the immunity. Within the cell's cytoplasm, the endonuclease dicer recognizes the double-stranded RNA molecules and cleave them into shorter siRNA molecules of length ranging from 21 to 25 nucleotides with a 2-nucleotide overhang at the 3' end. One of the strands of the siRNA duplex, known as the guide strand, is loaded onto an organoid complex. The organoid complex is at the core of an RNA-induced silencing complex known as RISC. The RISC is a ternary complex which comprises of three main components, namely an organoid protein, secondly a dicer, thirdly a double-stranded RNA binding protein which is known as TRBP in humans. The second strand of the RNA, known as the non-guide or passenger strand, during the process of loading is cleaved by an organoid protein. As you can see in the diagram here, I had told you the siRNAs are derived from longer double-stranded linear RNA molecules which then associate with dicer within the cytoplasm and are cleaved into shorter 21 to 25 nucleotide long molecules. These molecules are then loaded onto the RISC complex. The RISC complex, which comprises of three separate components, which you can see in three different colors: blue, purple, and orange. Now, since it is double-stranded, one of the strands is known as the guide strand, while the other is the non-guide strand. The non-guide strand undergoes degradation such that the RNA now becomes single-stranded, and this RISC complex engages in finding the target mRNA, which has a sequence complementary to that of our guide strand, so that the two can base pair. Upon base pairing, there is an endonucleotic cleavage, followed by degradation of the mRNA through the action of ribonucleases. Both the enzymes DROSHA and DICER belong to the RNase 3 family, which upon cleavage of double-stranded RNA produce small RNA molecules with termini containing a monophosphate group at the 5' end and a 2-nucleotide overhang at the 3' end. The microRNA sequence and its reverse complement upon transcription base pair, giving rise to a double-stranded stem loop structure called the primary miRNA structure, that is around 65 to 70 nucleotide long. The microRNAs are transcribed from basically two sources, either from their own promoters or from sequences within the intron of other protein coding genes. As you can see in the following diagram, the longer primary microRNAs are processed by the enzyme DROSHA along with DGCR8 into smaller pre-microRNAs. These pre-microRNAs then are transported outside the nucleus through the help of the protein exporting 5 through the nuclear pore complexes, where they reach the cytoplasm. Upon reaching the cytoplasm, these pre-microRNAs come in contact again with DICER, which removes the loop of the pre-microRNAs, converting them into mature microRNAs. These mature microRNAs then are loaded again on the RISC complex. Within the RISC complex, just like the siRNAs, they contain a guide strand and a non-guide or a passenger strand, only one of which will be subsequently used as a template for binding to the target mRNAs. The enzyme complex known as DROSHA DGCR8 is localized within the nucleus, and that is why it is able to recognize the hairpin loop structures which help them to process the primary mRNAs into the pre-mRNA molecules. The DROSHA is an RNase 3 enzyme, while DGCR8 is a double-strand RNA binding protein. In plants that do not express DROSHA, the processing of primary microRNA and pre-microRNA is done by DICER, probably within the nucleus, and the mature microRNA duplexes are then transported to the cytoplasm by the protein exportin 5. The plant microRNAs mediate the silencing of target mRNAs just like the siRNAs, whereas in case of animals, the microRNAs silence target mRNAs without slicing. The guide strand is loaded onto an organoid complex just like in case of siRNA, but due to partial complementarity, the arginate proteins do not slice the target mRNAs. Certain arginate proteins are also known to lack catalytic residues required for slicing of mRNAs. If we talk about the role of the guide strand of the miRNA in mediating RNA interference, then it is its 5' end that is probably responsible for matching and binding to the target mRNA, whereas the 3' end helps to physically arrange the target mRNA into a cleavage favorable RISC region. The complete mechanism of miRNA mediated silencing is still to be worked out, but has been proposed to take place by repressing the translation of target mRNA and removal of a poly-A tail, which causes the degradation of the mRNA. The organoid proteins are known to localize to specific regions within the cytoplasm, which are known as P bodies or cytoplasmic bodies or the GW bodies. These P bodies are regions characterized by high rates of mRNA decay as well as microRNA activity. Any kind of disruption of these P bodies leads to a decrease in the efficiency of RNA interference, thereby signifying its critical role in the RNA interference process. The PIWI interacting RNAs or the piRNAs are a third class of molecules that mediate RNA interference and are 24 to 31 nucleotide long. Their main function is to silence transposons in animal germ cells. Currently, both the biogenesis as well as the mechanism of action of piRNAs is not very well understood, but some understanding of this model has been worked in Drosophila melanogaster. The precursor molecules of piRNA are single-stranded RNAs, as the biogenesis of piRNA does not need the enzyme dicer, like it is used for the biogenesis of siRNA and microRNA. The small RNAs cause slicer-dependent cleavages of both the sense and the antisense transposon transcripts, which is facilitated by the proteins of the organoid family such as PIWI, the aubergine known as AUB, and the organoid 3 known as AGO3 in Drosophila melanogaster. The sense piRNAs are generated via slicing of sense transcripts by PIWI or AUB proteins. The piRNAs then associate with AGO3 to direct the slicing of antisense transposon transcripts. This generates antisense piRNAs, which in turn binds to PIWI and AWB and AUB proteins in order to guide the slicing of sense transposon transcripts for generating the sense piRNAs, and this process continues. We will now talk about the pathways that are involved in RNA-induced gene silencing. Now, either the siRNA or the microRNA molecules associate with RNA-induced silencing complex, although both arise from different sources. But before associating with RISC, both the precursors of siRNA as well as microRNA first associate with dicer, where they undergo processing and their size becomes smaller, around 20 to 25 nucleotide long. But after associating with RISC, one of their strand known as the sense strand undergoes degradation while the other strand known as the guide strand is retained. It is at this point that their mechanism of action undergoes a change. This change is mediated or is done due to differences in the nature of the target mRNAs. Now, there are certain mRNA molecules that show 100% sequence similarity to the Si or the microRNA that is present in the RISC complex. In this case, the RISC mediates endonucleatic cleavage of the target mRNA, thereby initiating degradation of the mRNA. In the second case, the target mRNA may not show 100% sequence homology to the RNA mediating gene silencing. So in this case, the mRNA does not undergo endonucleatic cleavage, rather it undergoes translational repression. And there is also a possibility that this mRNA is cleaved at its poly A tail, followed by action of ribonucleases which degrade this mRNA. The third function that these molecules do is to bring about a decrease in gene expression via the association or via the recruitment of chromatin remodeling enzymes to specific sites within the genome. But for this, instead of associating with RISC, they associate with RITS, which is RITS, which is RNA-induced initiation of transcription silencing. For this, the chromatin remodeling complexes cause methylation of the histones as well as certain DNA residues, thereby causing the formation of heterochromatin. This is the phenomena which is responsible for the formation of centromeres. Due to formation of heterochromatin, these regions are not simply transcribed easily. Thereby, all these three different phenomena, that is degradation of the mRNA, translational repression followed by heterochromatin formation, leads to RNA-mediated gene silencing. The following are the biological functions of RNA interference in our cells. RNA interference provides immunity against viral infections or the entry of any foreign exogenous DNA. RNA interference also mediates both the downregulation and the upregulation of genes. RNA interference is an important part of the immune response to viruses and other foreign genetic material in both plants and animals. In plants, RNA interference prevents the self-propagation of transposons. Arabidopsis thaliana has been reported to express several homologs of the enzyme dicer, each of which is specialized to react differently against different viruses. Certain plants, upon infection by particular bacteria, express endogenous siRNAs. In Drosophila, RNA interference provides antiviral innate immunity and also protects actively against pathogens like Drosophila X virus. In C. elegans, the organoid proteins and other components of the RNA interference pathways are upregulated upon viral infection, thereby conferring resistance to the worm. In mammals, as of now, the exact role of RNA interference in innate immunity is not well understood. RNA interference is involved in downregulation of genes. It was in the year 1993 in C. elegans where there was first time reporting of the role of endogenously expressed microRNAs in downregulation of the expression of genes. Similar function is performed by the jaw microRNA in plant Arabidopsis, where it regulates several genes that control plant shape. Both intronic and intergenic microRNAs mediate translational repression, regulate development, particularly the timing of morphogenesis, and maintains undifferentiated cell types like the stem cells. RNA interference is involved in epigenetic phenomena such as gene imprinting and X-chromin activation. RNA interference is responsible for the formation of centromeric structure in chromosomes. RNA interference pathways repress transcription in two ways. First, by targeting transcription factor mRNAs, and secondly, by reduction in the levels of specific transcription factors which automatically reduces transcription in genes, which automatically reduces the transcription of genes. In plants, the microRNAs mainly regulate the genes encoding the transcription factors and the F-box proteins. The abnormal functioning of microRNAs are known to be associated with the occurrence of diabetes, heart disease, tumor formation, and dysregulation of the cell cycle. Thus, we can conclude that the microRNAs are so important that they perform the dual function of oncogenes and tumor suppressors. The phenomenon of RNA interference is also involved in upregulation of genes. Yes, both siRNA and the microRNA having sequences complementary to the promoter can increase gene transcription, a phenomenon known as RNA activation. This occurs with the help of the protein enzyme dicer and the organoid proteins, which possibly cause histone demethylation. The following are the applications of RNA interference. As of now, not all of them have been implemented, but they're under different stages of development. The first is in the field of biotechnology, where RNA interference is used in basic research to study gene knockdown in transgenic plants, and as RNA interference is also being developed as a potential insecticide. RNA interference is playing an active role in developing medicine in the form of antiviral therapy and in treatment of cancers. The RNA-mediated gene silencing is a specific and inexpensive method to rapidly analyze the function of a gene, either in a cell or in a model organism. Simply for this, the double-stranded RNA molecules with sequence complementary to the gene of interest are synthesized and introduced into a cell or organism. There, it is recognized as foreign genetic material, thereby activating the RNA interference pathway, which causes a drastic reduction in the expression of a targeted gene. RNA interference is also known as gene knockdown because it does not completely abolish gene expression, unlike the gene knockout procedures where gene expression is completely eliminated. RNA interference helps in creating specific single gene defects without the need to create a transgenic organism carrying the mutation in its genome. Researchers using computational biology are trying to design double-stranded RNA reagents that can maximize gene knockdown but minimize off-target effects. These off-target effects arise due to the presence of repetitive sequences in the double-stranded RNA, which pairs with multiple mRNAs having sequence similarity, which leads to reduction in the expression of multiple genes. The exogenous RNA to be introduced can either be long, so it can be cleaved by the enzyme dicer, or it can be short, so it serves like an siRNA substrate based on the organism and the experimental design. In mammalian cells, the siRNAs are used because long double-stranded RNA molecules activate interferon response, which reacts non-specifically to exogenous genetic material. The mouse oocytes or the cells from early mouse embryos are used as mammalian model system for studying gene knockdown, as they do not show interferon response to exogenous double-stranded RNA. RNA interference has huge application in the field of crop sciences. Using RNA interferences, the researchers have developed nicotine-free tobacco, decaffeinated coffee, nutrient-fortified and hypoallergenic crops. The Arctic apples have been produced by RNA-induced suppression of polyphenol oxidase gene. This will prevent the apple from undergoing browning after being sliced, as they are unable to convert the chlorogenic acid into quinone. These apples will soon be receiving US approval. RNA interference has been used in plants to lower the level of natural plant toxins. For example, the cotton seeds contain the toxic compound gossypol that makes them unsuitable for human consumption. So efforts are on to reduce the concentration of this compound to make them edible. The technique of RNA interference have been used for reduction in the linamarin in cassava plants. Through the use of RNA interference, researchers have successfully reduced the levels of allergens in tomato plants. RNA interference has been successfully used in the laboratory to develop resistance to common virus affecting plants. RNA interference is being actively applied in the field of generation of transgenic plants also. Transgenic crops expressing RNA to silence important genes in target pests have been developed. Monsanto will probably be the first company to market the transgenic corn seed expressing double-stranded RNA based on gene SNF7 from a beetle called the western corn rootworm. The larvae of this beetle annually cause damage worth $1 billion in the United States. A study conducted in 2012 showed that silencing of this gene results in stunted larval growth, which kills them within days. Later, in 2013, the same team reported negligible effects of RNA effects on other species. RNA interference is being developed as an insecticide. Researchers are trying to incorporate the protein cry and RNA interference in one plant to delay the onset of resistance in insects. As of now, not much success has been achieved, as there is no negative effect of RNA interference on cotton bollworm, the beet armyworm, and the Asiatic rice borer. Use of RNA interference in different species of butterflies and moths have given varying results because their saliva is better in breaking down RNA, making the technique a little ineffective. RNA interference has an important application in the field of medicine. Research is on to develop RNA interference as a potent pharmaceutical agent against a disease caused either by overexpression of a gene or normal expression of a mutant gene. RNA interference in vivo delivery is easily accessible to surface tissue such as eye and respiratory tract. For example, siRNA were used in direct contact with the tissue where they were successfully focused on target genes. For delivering siRNA to deep tissues, it must be protected from nucleases, which makes targeting specific areas difficult. To overcome this problem, researchers tried to inject high levels of siRNA to ensure their delivery to deeper tissues, but this ultimately led to hepatotoxicity, making it a non-feasible approach. Surface application of siRNA molecules in animal models have proved successful in treating virus infections, eye diseases, cancers, and inflammatory bowel disease. Application of RNA interference for the potential treatments of neurodegenerative diseases like the Huntington's disease has been proposed. Using RNA interference in tissue cultures, there was a reduction in the severity of infection by HIV, influenza, and poliovirus. Thus, people are proposing to use RNA interference as an antiviral RNAi therapy. Potential antiviral therapies include treating infection by herpes simplex virus type 2, inhibition of viral gene expression in cancerous cells, knockdown of host receptors and co-receptors for HIV, silencing of influenza genes, hepatitis A and B genes, inhibition of measles viral replication. RNA interference technology is used to silence overexpression or abnormal expression of key proto-oncogenes in tumor cells. Different types of cancers have differential expression profiles of microRNA, which can be used for diagnosing tumors, predicting their course, and planning of treatments. In some cancers, there is a defect in microRNA gene expression, which can be treated by injecting synthetic siRNAs. Safe delivery methods for transporting RNA molecules are required, but presently viral vector systems are used, but due to safety concern, methods like liposomes are being considered. A lot of progress has been made in developing RNA interference pharmaceuticals. This involves completion of phase 1 of the first human clinical trials, completion of phase 2 and phase 3 clinical trials for using siRNAs to treat age-related macular degeneration caused by expression of the vascular endothelial growth factor gene. Clinical trials to test the use of RNA interference for treating hepatitis B and respiratory syncytial virus infections have begun. Clinical trials are planned for using siRNA to treat influenza and hepatitis C virus infections and some solid tumors. In mouse models, RNA interference is proved to be effective in reversing induced liver failure. So students, let us now summarize what we have learned in this module. RNA interference is a biological process where the RNA molecules cause gene silencing by either initiating mRNA degradation or mediating translational repression or carrying out genome modification. RNA mediating gene silencing was first discovered in plant Petunia, but the underlying mechanism was not known. It was Fire and Mellow's work which led to the discovery of RNA interference in C. elegans. The RNA interference is mediated by a double-stranded RNA that shares sequence homology with the mRNA or the gene they target. RNA interference is majorly mediated by siRNA and microRNA. The siRNA is derived from longer, linear, double-stranded molecules located within the cell's cytoplasm. The enzyme dicer cleaves the double-stranded RNA into 21 to 25 nucleotide long siRNAs with the overhang of 2 nucleotides at the 3' end. The guide strand of siRNA is loaded onto an organoid protein, which is a component of RNA-induced silencing complex known as RISC, while the non-guide strand is cleaved by the organoid protein. The guide siRNA then associates with the target RNAs containing perfectly complementary sequences, which are subsequently sliced. The microRNAs are short RNA molecules transcribed from the sequences within the introns of their own protein coding genes or from their own promoters. The microRNA, upon transcription, has a double-stranded stem loop structure called primary microRNA structure that are some 65 to 70 nucleotide long, which is subsequently processed to pre-microRNA by the DROSHA DGCR8 complex. The pre-microRNA is then exported to the cytoplasm by exportin 5, where the enzyme dicer cleaves it into a 20 to 25 nucleotide long linear double-stranded microRNA. The guide strand of microRNA is loaded onto an organoid protein of RISC. The microRNAs are partly complementary to sequences at the 3' untranslated regions of their target mRNAs. Hence, the organoid protein does not slice the target mRNA. The piRNAs are the third class of RNA molecules mediating RNA interference. They are approximately 24 to 31 nucleotide long and silence the transposons in animal germ cells. The biological functions of RNA interference include protection against viral infections, upregulation and downregulation of genes. Applications of RNA interference are in the field of basic research, biotechnology, medicine, agriculture, functional genomics, and the list is almost endless. Thank you.